Good morning dear students and welcome. We will be studying about cell, the chapter cell, and this is class 10 and we are strictly following Concise biology class 10. So the first thing that we have an idea, or that I would like to ask you, that what is cell, right? So we have already learned about cell and cell organelle in class 9 in your previous class. So today we will be starting about the cell and the chromosome. What are chromosomes, or what we are doing about, or how is cell division occurring. Okay, so the first concept that we will be studying here is structure of chromosome. Now you all will ask me that, sir, what is chromosome, right? Coming to the fact of what is chromosome, the first let us quickly revise what is cell. So cell is nothing but the structural and functional unit of a body. Now what does this mean? The structural and functional unit of body. The structure, I hope it is visible to you all, right? So what does it mean, structural and functional unit of body? That means, suppose we can give it as example of for example, we are building a house. Okay, so when we are building a house, the first thing that came into our mind that should be used while building a house, that is nothing but a brick, right? So we can call brick as the structural unit, or is the smallest unit which builds up the whole house. So when we will be putting that exact concept in biology, we will find that our whole body, be it human, be it any plant, or be it anything, the whole body is made up of what? Is made up of cell. Now this goes for the concept of structural part. What is the why did I told as the functional part also? Can you all answer? Okay, no. See, why we call it as a functional part also? Because how our whole body functions, that particular thing, that particular each and every function is also the function of the cell. I repeat, how our body are performing, how our body are functioning, that each and every particular thing our cells are also doing that. And that is why in our previous class, in class 9, while we were studying about the cell, the basic structural and functional unit of our body, we have already learned there that each and everything, like our organs work, we call our cell organ as cell organelle, right? So cell is a structural functional unit of body. Now we will go for chromosomes. What are they? Now students, see, as chromosomes are highly coiled and condensed chromatin fibers. So we can define chromosomes as they are highly condensed and coiled chromatin fibers. Now you all will ask me, what is chromatin fiber? Sir, you have told that it is highly condensed and coiled chromatin fibers. Now what are chromatin fibers? Okay, so I hope you all are also following and your the book is open in front of you. Okay, because see, you need to keep your book in front of you, as this is an online class going on, right? So I will be giving some figures, some descriptions from that particular chapter and particular page number. So as of now, please open Concise Biology Part 2, that is class 10, and page number 8. What are chromosomes? Okay, the nucleus contains most of the cell's DNA, which is organized into discrete units called chromosomes. So the nucleus, suppose we can call suppose we can call it as a cell. Okay, now if we call it as a cell, I hope it is visible. Yes, it is visible. So if I call it as a cell, Now there are several cell organelle present here, say these, there are several things. Now including it, there is a nucleus. Okay, now what is this nucleus? This nucleus is present inside the whole cell. For this particular drawing or diagram, you can refer to figure 2.1 on page number 9. Okay, I am writing here figure 2.1, page number 9. It's ICSC Concise Biology, class 10. Okay, so if we see there, here we will find that there are, suppose, very lightly, I repeat, very lightly fiber like shapes, which we call them as chromatin fibers, Now what are chromatins? Each chromosomes contain one long DNA molecule associated with many protein. Okay, now what is DNA? Do any one of you know what is DNA, or what is the full form of DNA? Now for DNA, see, we call it as a DNA. Now what is DNA? DNA is nothing, I repeat student, DNA is nothing but it is a double helical. We'll study later on. In this particular chapter we have DNA. It is a double helical structure of protein and protein complex. Okay, now DNA, the full form of DNA is deoxyribonucleic acid. Okay, now when this DNA along with other protein substance are present inside the nucleus, we call them as chromatin fibers. Okay, we call them as chromatin fibers. Now these chromatin fibers, when they are in the cell and we dye it with particular dye material and see it under the microscope, we find they are very lightly placed, they are very lightly substance, and they appear like a fiber shaped. Okay, now what happens during cell division, these chromatin fibers, when we observe under a microscope, we found that these chromatin fibers becomes thick and condensed, and when they are dyed with color, they are found to be very much condensed and thick inside the particular cell's nucleus, and that chromatin fibers, which are highly coiled and condensed, we term them as chromosome. So I hope up till this, what is chromosome and what is chromatin fiber, up till this the things are clear to you. So for any of the questions you can definitely ask me, but now going on to what are chromatin made up of. Okay, so let me clear it up quickly. Now what are chromatin made up of? Now chromatin fibers are made up of two parts. One is DNA, as I already told you. Another one is histones, Histones. Now what are histones? Histone is a particular type of protein. Okay, you can follow page number 9, there it is written chromatin, and it is made up of DNA and histones. Okay, so DNA is deoxyribonucleic acid, and it contains about, this DNA contains about 40%. Okay, and histone contains about 60%, my dear student. So DNA is of 40% and histones is of 60%. Okay, now molecular structure of DNA. What are the molecular structure of DNA? So molecular structure of DNA you will find in your book. It is given figure number 2.2 in page number 9. You will see over there. The thing is, it is like this. It is a double helical structure. Okay, so this we can call as a schematic diagram of helical structure of DNA. Okay, let me quickly write here DNA. Now, what are these? What are these strands? You need to understand. You might have questioned me, sir, what are these? What are these helical structures and all the thing? Now, the shape of DNA molecule was first studied by Rosalind Franklin in 1953. However, the structure was finally worked by Watson and Crick in the same year. So, we call it as a Watson and Crick model. Okay. A single DNA molecule is very large and hence is described as a macromolecule. Okay. This is described as a macromolecule. Now, each single DNA strand composed of a repeating nucleotides which are made up of 3 components. So, let me quickly come here. Study about DNA. Okay, the same thing, this is the same chapter. Okay, everything is going under this particular chapter, chapter 2, structure of chromosome, cell cycle and cell division. So, DNA. Okay, now each single DNA strand is composed of repeating nucleotides. We will come to this. Okay, wait. You will always understand by a bit simpler form. Okay, now this is what? This is the repeating strand. Okay, this is made of the repeating strand. Okay, now along with this each single DNA strand is composed of repeating nucleotides. These nucleotides are made up of 3 things. What are they? The number one is they are made up of 3 components. The number one is phosphate. Phosphate. Okay. Number 2, it is made up of sugar. I hope what I am teaching you all is understandable and you can go through this. Okay. One is phosphate, another one is sugar, and the third part is a nitrogenous base. Okay, now this video I am making exclusively for class 10 because we all know that this pandemic is going on and we are staying in our house, and me being teaching for more than 6 years, I know that what students are facing today in the today's scenario. There is no proper classes going on in the school, as well as there is no proper tuitions going on. Everything is going in online mode, and yet we need to study, right? We need to study. Whatever happens, we need to understand, we need to keep our concept clear, because as the board exams are just nearly 9 to 10 months knocking in your door, so we need to be advanced and prepared so that we can fight with the board exam as well while fighting with corona. So coming back, the nucleotides, it is made up of, see, DNA, this DNA structure is made up of nucleotide. These nucleotides are the repeating strand which consist of the DNA. Now this nucleotide is made up of 3 parts. One is phosphate, one is sugar, another one is nitrogenous base. Okay, so this is the thing. Now please follow your book, page number 9. There you will find page number 9, on the second column you will find that each single DNA strand is composed of repeating nucleotides which are made up of 3 components: phosphate, sugar one is pentose. Okay, so let me write here, it's pentose, and these two are arranged lengthwise. Okay, and a nitrogenous base, this nitrogenous base is attached to the sugar inward. It is attached to the sugar inward. Okay, it is attached to the sugar inwards, which extends to join by a hydrogen bond the complementary nitrogen base from other strand. Thus, now these two are joined by a hydrogen bond. I hope you all know about hydrogen bond. What are they? This is joined, this sugar and nitrogenous base. Nitrogenous base is joined with sugar inward with the help of a hydrogen bond. Okay, hydrogen bond. Okay, now moving forward, we will find that thus the two stand together make a ladder-like arrangement with the nitrogenous base forming the rungs of the ladder. 4 types of nitrogenous base are found in a DNA. Now, what are the type of nitrogenous base? I am removing the structure. We'll go on it later on. So nitrogenous base are of 4 types. 1, 2, 3, 4. Okay. The first one is adenine. ADENINE. Okay, we can call it as a E. The second one is guanine. We call it as a G. The third one is cytosine. We call it as C. The fourth one is thiamine. We call it as a T. So what are they? They are adenine, guanine, cytosine and thiamine. Okay, I hope these are the part of nitrogenous base that is found inside DNA. I hope this is clear to you. Now, one more thing that you need to understand here is that adenine pairs with thiamine with 2 hydrogen bond. Adenine pairs with thiamine. Okay. Thiamine. This pairs with this with 2 hydrogen bonds. Okay, we can call it as a AT. This is one. So this A, this T, and this is with 2 hydrogen bond. Okay. Guanine pairs with cytosine with 3 hydrogen bond. Now these two pairs with each other with 3 hydrogen bonds, and we can call it as a GC. You can call it as a GC. I hope you all are being able to understand what DNA is. Also, we write it, quickly look here, for short we write it as A double bond T, and for GC we write it as G triple bond C. Okay, so this is all about DNA. This is all about DNA, and I hope up till DNA everything is clear to you. Now we will move on to histone proteins. What are they? As I have already told you that chromatin is made up of two parts. One is DNA, another one is histone protein. So we'll move on to histone protein. So now we will study about histone proteins. What are histone proteins, right? There is a question for you all. So can you tell me what are proteins? Okay, it's a homework for you all. You need to tell me what are proteins. Okay, now histone proteins, you can see page number 9 of ICC biology. There you will find that histone proteins. Histone are proteins that help in coiling and packaging of the DNA into structural unit called nucleosomes. So one important term that we get here is nucleosome. What are nucleosomes? Nucleosomes is, see, let me make it a bit clear to you all so that you can understand. The thing is that the DNA strand wind around near a core of 8 histone proteins. Okay, so this DNA strand that we have made earlier, that now these have a core of 8 histone proteins, and histone proteins, each such complex is called nucleosome. Okay, a single human chromosome may have about millions of nucleosomes. So see, one thing you need to understand, that the chromatin fibers or the chromatins that we have, these chromatins are of two types, as I've already told. One is the DNA part, another one is the histone protein part. Now these histone proteins helps in the, say what I call, this histone protein helps in the packaging and making the part like to stay the structural part of the particular chromatin fibers. Okay, so you can see, you can find it here. Histones are proteins that help in coiling and packaging of DNA into structural unit, and these structural units are called nucleosomes. These structural units are called nucleosomes. Okay, so a single human chromosome may have about 1 million of nucleosomes, and you know, like once one of my students asked me while I was teaching that, sir, how many nucleosomes do you have? Now I questioned that particular student that, can you tell me that how many say cells do you have? So it's a question for you all as well, that how many cells do you have, and that depends how many nucleosomes we have, how many chromatin fibers we have. Okay, another thing, that these nucleosomes or the chromatin fibers, they're highly coiled. Okay, they are like this structure, they're coiled, and see the coils that I am making right now, if I would have made it in a straight line, then it would have crossed this board for at least 10 to 20 times. But see, this particular coils is contained only in this part of this whiteboard. So the main thing for doing this coiling purpose is just of to put a long strand of DNA in a very small place in a coiled manner. Okay, the next part we will go to structure of chromosomes. Student, I hope that you all are being able to understand what I am teaching you. For any of the clarification or any of the queries, you can definitely ask me. These are the structure of chromosome. So up till now, I think what are chromosomes, what are chromatin fibers made up of, these everything are clear to you. Now we'll go to structure of chromosomes. See, they are made up of chromatin fibers, as we all know. Suppose I am drawing it like a structure of chromosomes. Okay, though it is not actually a chromosome and it looks very funny, but see, fun is also important in a class. Otherwise, if I go on teaching you all from the particular chapter, going on reading, then it would have no use. You all will be in a state of very shock that what biology teacher is teaching us. We are not been able to understand each and everything. Like I am teaching you each and everything, but you all are not getting a single point of me. Then what is the point of teaching you all, right? So this is a let us assume this to be a structure of the chromosome. Let us assume this. Okay. Let us assume this for a structure of chromosomes. Now, what we will find is these parts are known as chromatids. Okay. Also, you can follow page number 10, structure of chromosomes, figure number 2.5 and figure number 2.6. This point of attachment is known as centromere. Okay. Now, each chromosome in its condensed form is visible during the start of cell division. Okay. Otherwise, without this condensed form, these chromosomes are not that much visible. Okay. We can call it not that much visible. Anyway, so each chromosome in its condensed form is visible during the start of cell division, consists of two sister chromatids join at some point. Okay, these are two sister chromatids joined at some point, and this is known as centromere. And it appears as a small constrictor region. Each chromosome centromere is located at a particular site. The centromere also serves as a point of attachment, serves to attach the spindle fiber during cell division. Now we will come to it later on, what are spindle fibers and how cell division occurs. Okay. Now, after completion of the cell division, the chromatids decondensed and revert to their very long fine thread like chromatin fibers. These would be as many chromatin fibers inside the nucleus as the number of the chromosome that appear during cell division. So what happens is that when these particular chromosomes are ready for cell division, or they go for cell division, they become thick, condensed, and we call them as a chromosomes. Otherwise we call them as a chromatin fiber. Okay. Now, when they are ready, they have already divided, cell division already occurred. What happened when cell division already occurred? What happened? They become decomposed, they become decondensed again. Okay, and this is known as, or this we will study later on. Okay. So this is up to structure of chromosomes. Another point before going for the need and everything, what are genes? So can you all tell what are genes? Genes are specific sequence of nucleotides on a chromosomes that encode particular protein which express in the form of some particular features of the body. Okay. I have given you the definition of what are genes, and it is also available in the book. But my concept, or the thing for which I am teaching you all, is what are genes. See, the next chapter is of genes. We will definitely get that. Now, particularly, we have learned a lot, or we have heard a lot about saying our father or mother that you have got typical characteristics from your father. Or we have already get a point that we have already heard that our mothers are saying, this is from your father's side. This particular thing, this particular bad behavior is from your father's side. Or your mother saying your father that, what are you? You haven't teach anything to your son or to your daughter. Now, also these are all fun apart. We have also studied that, or we have also observed that any of your character, be it physical character, be it emotional character, be it anything, that comply with your genital or compile with your father's side or your mother's side. For example, your looks. Suppose it is, we can say that it resembles your father, it resembles your mother. So what are all these, or how are they carried forward? They are carried through this particular gene. I repeat, they are carried through their particular gene. So when you will call about what are genes, so genes are specific sequence of nucleotide on chromosomes. The nucleotides these are nucleotides. So these nucleotides, what we have what we have learned about nucleotides? Nucleotides are made up of nucleotides are the structural or the packaging part of chromatin fibers, right? So here we will find that nucleotides, the part which is inside nucleotide, and it is the specific sequence of nucleotides. It is specific sequence how these nucleotides are being made up of, how the structural part is made up of. See, I have already told you this coil, like coil. I have already told you this coil thing, but how this coil is being made, how this particular arrangement of histone proteins, DNA structures and everything that is inside these nucleotides, it is a sequence that is known as the genes. I hope this is clear. The genes are specific sequence of nucleotides on a chromosomes that encodes particular protein which express in the form of some particular feature of the body. Okay, so these are genes. So one more thing which is not included in your ICSE syllabus, that is DNA fingerprinting, but yet each and every individual have unique set of DNA or unique structure of DNA. I repeat, each and every individual have unique set and unique structure of DNA, and we can identify on basis of that. So all of you have heard about fingerprinting, all of you have heard about what are fingerprinting. So DNA fingerprinting is nothing but to study the structural shape and the constitutional shape of particular human or of a particular living being is known as DNA fingerprinting. Now these DNA fingerprintings are nowadays very much used in forensics, in criminal cases, to find the convicted person, to give judgment. These DNA fingerprintings are used because, see, it may happen not always we leave a fingerprint, or not always fingerprints are available, but DNA fingerprinting is more accurate than normal fingerprinting. Okay, so what are the need for new cells? The need for new cells, we all know that, is already written in your book, is for growth, for replacement, for repair, for reproduction. Okay, one part that I will make you understand, or I will explain it in a bit more way, that is for reproduction. We will also go through this in the later on of the chapter. But for reproduction, see, cell division, there are two types of cell division. I repeat, cell division, these are of two types. One is mitosis, another one is meiosis. Excuse me. Okay, so what are mitosis and what are meiosis? See, when the cell divides from its own, when the cell divides from a single parent, I repeat, when a cell divides from a single parent and forms a new offspring or a new daughter cell, we call them as mitosis. But when this thing happens in meiosis, we call them, when this thing happens where two mother cell or two parent cells are incorporated and one new organism or new cell is formed, we call them as meiosis. Okay, so see, can you all tell me that what are the number of chromosomes that are present in human? Okay, that's 46. See, 46 pair, 46 Okay, 46, that is 23 pairs. Now these 23 pairs or 46 chromosomes are present in a man or in a human. So in meiosis, what happened, and here you will find it, page number 11, point number 4, for reproduction. What you will find is that it is given here 23 pairs here, that is 46. Now this 46 we can write it as 23 pairs means into 2. Now here if we take 23 as n, we can write it as 2n. We can write it as 2n. For meiosis or for reproduction, what happens? Two parents, as I have already told you, the two parents are needed, right? See, this is first parent, this is second parent, these have 2n cell, this particular second parent also have 2n cell. What happens? N cells come from here, another n cell comes from here, and it comes n plus n equals to 2n. So we called meiosis as the involvement of two parents, and this is known as meiosis. And in mitosis, what happens? If there are say 2 chromosomes, it breaks down to 1, or a single strand gets break down. A single strand, see, this is a single strand, this breaks down from here and it becomes 2 new individual strand. Okay, so this is what we have all studied about up till the first part of the chapter. Okay, so on my next video or in my next lecture, we will continue about cell division. What are they? What are the two types of cell division that I have already given you a brief about cell division, mitosis and meiosis. We will study about what are cell divisions, how they occur, what are their types and everything. But up till today, I hope everything is clear to you. For any of the queries, please do tell it. I will try to resolve it. Study hard, and thank you for getting understood this chapter. This is a very important chapter to study seriously. Thank you.